Today's session on psychedelic neuroscience is really a remarkable confluence of this phenomenological and neurobiological perspectives. We have three outstanding speakers, and I would like to welcome our first speaker, Robin Carhart Harris, who has been doing pioneering work in the neurobiology of the psychedelic experience. Robin.
Hi everyone. Thanks, George. It's a real pleasure to be here again. It's my third time in Tucson. I was here in 2012 first and 2016 as well. So it's lovely to be back. I do love it here. And it's nice to be telling you about my favorite topic, psychedelic drugs, how they work in the brain, and what they can tell us about consciousness. Here we are. So, psychedelics, what are they? Let's start with the basics. I've listed three important serotonergic classic psychedelics here. Psilocybin and its metabolite psilocin, 4-hydroxydimethyltryptamine, discovered in the late 1950s by Albert Hofmann and used for millennia by certain cultures in ceremonial healing contexts. Dimethyltryptamine, discovered in the early 30s by a German chemist and then found in plant material used by certain cultures in the late 1950s. Who were using ayahuasca, this Amazonian plant brew. DMT is found in over 50 species of plants and a few species of animals as well. It's also been found in our bodies, as Jimo will tell you about after me. And it's been coined as the spirit molecule by Rick Strassman. It's something that we're studying at the moment, and I'll show you a little bit of data on the effects of DMT on the human brain later on. LSD, the prototypical psychedelic discovered by Albert Hofmann again, 1943. Its psychoactive effects discovered then and used in psychiatry since the early 1950s. Another compound that we've studied with functional brain imaging. Now, if you look at the molecular structure of these classic serotonergic psychedelics, one thing that's striking is how similar these structures are to an important neurotransmitter in our brains. In fact, most of the serotonin in our bodies is actually in our gut, some 90%. It was discovered in the brain in the early 1950s, and then soon after that, it started to be linked to psychological health, mental health. It was found that LSD interacted with it. This was one of the reasons why academic psychiatrists, biological psychiatrists started to think that serotonin could be involved in different psychopathologies and perhaps potentially used in their treatment as well, because LSD has such profound effects on consciousness and was being used in psychotherapy throughout the 1950s into the 1960s. And it started to be looked at again today. So what about the basic pharmacology of these classic psychedelics? Well, these compounds work on the serotonin system. Essentially, they mimic serotonin. They kind of hijack the serotonin system. Serotonin system is arguably the most complex neuromodulator in the brain. It has some 14, at least 14 different receptor subtypes. Psychedelics, the idea and some evidence that they interact with the serotonin system first appeared in 1954, and then into the 60s it was hypothesized that they stimulate the serotonin system. Then into the 1980s, a key finding was that the affinity of a psychedelic compound for a particular serotonin receptor subtype, the serotonin 2 receptor subclass, later found to be the serotonin 2A receptor was found to be the key receptor at which psychedelics work. But this finding in the mid-1980s was that the affinity for the serotonin 2A receptor correlates very strongly with the potency of these compounds. So the higher the affinity, the higher the binding, the stickiness of a psychedelic for this particular receptor, the more potent the psychedelic. So LSD, for example, very high affinity and very potent. You only need very small amounts of it. So this has been a staple finding, really, in psychedelic science, if you want. It's also been substantiated, this rule, this principle that the serotonin 2A receptor is important by blocking studies, giving a serotonin 2A receptor antagonist before giving a psychedelic. This has now been shown with LSD, psilocybin, and also ayahuasca, that if you give a serotonin 2A receptor antagonist before a psychedelic, essentially you'll abolish the signature psychological effects of the psychedelic. So it's further reliable evidence that this receptor is key to how these compounds work in the brain. If you block those receptors, you won't trip, essentially. So where are these receptors, these 2A receptors? Well, the best evidence that we have for where they are in the human brain has quite recently come out using a quite selective serotonin 2A receptor agonist, using an agonist in the context of PET imaging is quite useful because it allows us to label receptors in their high affinity active state. And so this has been an important development recently, and it's been able to give us these nice images like this one that you can see on the left of where the serotonin 2A receptors are most densely expressed in the human brain. You can see that they're heavily expressed in the cortex, comparatively little in subcortical regions, and also the expression is highest in highest level aspects of the cortex. So those of you who are familiar with the default mode network will see that the expression is particularly high in key nodes of the default mode network, like the posterior cingulate cortex, you can see in this midline section, and also into the medial prefrontal cortex. So this might be a hint about how these compounds work on the brain, modulating consciousness so profoundly as they do. But if we just slow down for a minute and think about, rather than putting all our focus onto psychedelics, think about the system that they modulate, we can ask this very important question of what is the function of the particular receptor that they activate. And as I'll show you in a moment, there's a large amount of evidence that this receptor is involved in what you might call functional plasticity. So this would include neuroplasticity, but also any aspects of behavior and also subjective experience that you could link in with this umbrella term of plasticity, which could be defined quite simply as the ability to change. So there's a lot of studies cited here, but that's important because there's a lot of evidence to back up this principle that not just the serotonin system is important in modulating how we interact with our environment and how, crucially, how sensitive we are to our environment, but more specifically that the serotonin 2A receptor, this subtype that psychedelics work on, is vitally important in terms of modulating sensitivity to the environment. So some evidence to back that up. It's been linked to learning and also extinction learning or unlearning, if you want. Serotonin 2A receptor agonism, which essentially means stimulation, has also been linked with neurogenesis, particularly in the cortex. Also, the expression of the serotonin 2A receptor is highest during our lifetime, during critical periods. It's highest during our youth, and then drops into older life. You can see some PET images here with the heat maps there showing that the expression is very high in a 21-year-old and drops right down in a 68-year-old there. Also that 2A receptor stimulation or agonism has an interesting effect on the organization of systems in the brain. So during normal development, you'll see that systems differentiate, and that allows the brain to specialize. But what has been found with serotonin 2A receptor manipulation is that if you stimulate this receptor, it will promote a kind of inverse of that development, so a de-differentiation or a desegregation in the brain. It's the opposite of what you ordinarily see in a developing brain. So there's a kind of functional regression that's induced by activating this particular receptor. Sticking on this theme of the importance of the serotonin 2A receptor for interaction with the environment, also some genetic work has shown that a particular genetic variant linked to higher signaling at the 2A receptor mediates environmental sensitivity. So for example, if you look at early life stress, or the inverse of that, an enriched early life, then the particular polymorphism or genetic variant that's linked to more signaling, those people will be more sensitive to those differences in one's environment in terms of them being a predictor of later life outcomes like mental health outcomes. So again, further evidence that this receptor is important in terms of modulating our sensitivity to our environment. Also reiterating this point that early life stress also increases the functionality of serotonin 2A receptor in terms of increasing its expression, but also its signaling. If instead of looking at early life stress, we look at stress manipulations in general, so inducing stress, this effect also seems to promote serotonin 2A receptor functioning. And it also seems to mediate an animal's behavioral response to stress. So these are important principles that I'd like you to keep in mind, because we'll kind of go full circle here and come back to this theme towards the end of this talk. But if we segue now into this question of what psychedelics do to the human brain in terms of brain function as seen through the lens of functional brain imaging, I'll just show you a couple of images here that speak to a couple of important principles that we found. And one of these is that psychedelics appear to disintegrate systems in the brain, so brain networks, for example. We've seen this in a number of studies now with a number of different psychedelics, and other teams have also replicated these findings. And I'm showing an image here on the left of a functional connectivity analysis using the parahippocampus as the seed here, looking at how that region speaks to the rest of the brain, how correlated activity is in that region to regions in the rest of the brain. You can see a blue blob there, that's the retrosplenial cortex, posterior cingulate cortex, a key node of the default mode network, as the parahippocampus is, and you can see that there's a disconnect here. And you can also see that the magnitude of that disconnect or decoupling correlates very strongly with the volunteers' ratings of ego disintegration. So there's an interesting mapping here between phenomenological experience, subjective experience, and what's going on in the brain. So we see a functional disintegration in the brain, and people experience a functional disintegration in terms of a sense of their self dissolving or disintegrating. This is important because it gives us an important clue about how the brain works and what systems might be important for maintaining our sense of self or ego. And so the focus, the spotlight is put on this particular system, this particular circuitry, and it's highlighted here as an important candidate as a substrate of the sense of self or ego. If we look at the image on the right, this is more looking at integration, disintegration in the temporal domain. Here we're looking at a frequency spectrum and more specifically the effect of DMT, intravenous DMT given to 12 volunteers while we recorded their brain activity with EEG. The injection came where that vertical red line is. You can see the short section at baseline with the alpha power being very prominent. That's the yellow bar you can see. So as ordinarily you'll see in the brain during rest, especially with eyes closed, there's this very dominant alpha frequency. We inject the DMT, people are shot into this highly unusual DMT state where they report seeing entities, seemingly sentient beings, and it's highly unusual. And then if you look at the oscillatory activity in the brain, we see these marked changes. You can see that alpha band essentially collapse, it disappears, which is quite remarkable. Alpha is a curious frequency because it's so pronounced in humans and becomes more pronounced as we mature into adulthood. So a bit like the default mode network, it's perhaps one of the signatures of at least the capacity to have high-level cognition or consciousness as humans have. So it's intriguing again, and it highlights another potential candidate in terms of a aspect of brain function that may be crucial for the maintenance of our sense of self and the maintenance of normal waking consciousness. So you see that drop of the alpha rhythm. And what's curious, you can see this white trace here. That's actually showing the intensity of the drug effects. The peak effects were touching on 10 out of 10. We just used simple 0 to 10 ratings for intensity. So the peak effects are felt at about 3 minutes, and then they subside. And as they subside, back comes the alpha rhythm. So there's this nice temporal mapping between subjective experience and what's going on in the brain. So another principle to complement disintegration, modular disintegration if you want, if you can think of networks and systems, however spatially distributed and long range they are, as being modules. So to complement this principle of modular disintegration is a global integration, or a modular or system level desegregation. This is something that, again, we've seen with different psychedelics now, and different imaging modalities as well. I'm showing an image here on the left with psilocybin. Here you can see different communities of brain regions. The regions are the dots, the communities are the colors, speaking more freely to each other under psilocybin, under the magic mushrooms, if you want. A marked change from what you see during normal waking consciousness with placebo. Then if you look at another resting state functional connectivity analysis here on the right, the seed here is V1, it's the primary visual cortex, identified using retinotopic mapping. And you can see at baseline most of the aspects of the brain that will be functionally correlated, connected with V1 will be other parts of the visual system, showing that this system is quite discrete, it does a particular function, it does vision, and that allows us to have perhaps precise, crisp visual perception. Now you can look at the brain under LSD and you see this massive expansion in functional connectivity, much more of the brain contributing perhaps to visual processing under LSD. This effect also correlated, importantly, with the volunteers' ratings of complex visual hallucinations. So this means seeing quite complex imagery such as landscapes or beings with their eyes closed. Crucially, they had their eyes closed during these scans. So here are two principles: disintegration and also de-differentiation or desegregation in the brain. Now, complementing this is another principle which I'll pretty much run with now until the end of this talk, and it's this, I think, quite simple idea, and arguably too simple. Perhaps people can pull me up on that and we can debate it. That the complexity of brain activity, however we measure it, is related to the complexity of our subjective experience. Another way to term this is that the richness of brain activity correlates with the richness of subjective experience. So the principle is that it's a very important index of richness of conscious experience, perhaps conscious content, if you want to understand it in that way. So this is a principle that I introduced in 2014 and recently have revisited in a review paper just published in Neuropharmacology. You can see this schematic there where I identify a particular band in a spectrum of the level of complexity or entropy, if you want, in which consciousness is possible, and that there's a thin band in which you might identify normal waking consciousness, and different pharmacological manipulations can shift the quality of consciousness within this range. So, for example, sedatives or anesthetics will reduce the complexity, the richness, the entropy of brain activity. They'll simplify it, they'll make it more predictable, and so consciousness will become more predictable. It will reduce in content to the point that in the end, consciousness is lost and we fall asleep yeah. But there is also this possibility that consciousness could be manipulated in the other direction, that activity could be enriched and conscious experience could be enriched. And this is something that we've seen with psychedelic drugs quite recently, and this is something that I know Anil, Seth will talk about in more detail in his talk within this session. So here you can see a nice image looking at sleep from study in 2015. You can see the level of complexity or entropy on the y-axis there. You can see it's high level during wake, it dropping during NREM sleep, dreamless sleep if you want, and then coming back to near normal levels during REM sleep or dream sleep. You can see the traces there, and it's quite a nice illustration, if you want, of this principle of brain activity being rich during normal wake and then becoming more predictable during deep sleep and then becoming enriched again during dreamful sleep. And as I said, different manipulations, pharmacological manipulations, such as the giving of anesthetics or sedatives, have been shown quite reliably to decrease signal complexity. And we've recently shown that psychedelics will tune the brain in the other direction, enriching brain activity and enriching consciousness in parallel. So it's quite a simple principle. Let's see if it holds up as we go into the future. It's a principle that was actually born out of thinking about the brain in a thermodynamic sense. Our initial brain imaging with psilocybin revealed drops in brain blood flow in regions that are normally hyperperfused or particularly perfused, receive most or a lot of the brain's blood flow during baseline levels. So I sort of toyed with the idea that perhaps there's a more even spread of blood flow in the brain under psychedelics, and my friend Robert Leach, former colleague who I was talking to, said, like an entropic brain, and so the theme kind of stuck. And then also speaking with Carl Friston and his work around prediction coding and brain function, and crucially his free energy principle, put me onto the scent of thinking more about this principle in the context of information theory. And that's something that now really kind of takes the focus of this principle. And so this principle that entropy is an index of uncertainty, of unpredictability. It also tells us something about information content. And here the extension of this idea is that it indexes the richness of conscious experience. It's increasingly well supported by us in different studies, but also by other teams. Ayahuasca, for example, some fMRI data finding increases in brain entropy. And then this intriguing theme that I'd like to share, and I've shared in this recent rewrite of the original paper, that what could this tell us about different disorders, both in psychiatry and in neurology? Perhaps it's a challenging idea, but what if in disorders of consciousness where there's a pathological reduction in conscious content, you might, at least conceptually, be able to give a psychedelic and modulate the content of consciousness? It's something perhaps worth trying. People try deep brain stimulation and with some success, so perhaps we should think seriously about this idea and test it. Also, in the context of psychiatry, it's been long hypothesized that disorders that you might characterize as being, at least in the colloquial sense, chaotic, most typically psychosis, might exhibit a particularly chaotic quality of brain activity. This is something that's been written about much, but there's a nice review by Fernandez et al. In 2013 that shows that there is some evidence to back up this idea, and that some of the findings that don't fit the rule can be explained by things like medication status, also symptom status, and also age. So it's something that I'm keen to explore further as we look at psychedelics in the context of different psychiatric disorders, and how they might be used therapeutically, as well as tools to inform perhaps on the pathophysiology of certain psychiatric disorders like acute psychosis. So I recently just hinted to this link between psychedelics and their therapeutic application, and this is where I'll focus now. So we quite recently looked at psilocybin with parallel psychotherapy for the treatment of treatment-resistant depression, so quite stubborn depression. And we saw quite positive outcomes in that study. But in this talk, I don't really want to focus too much on that trial, but rather what's going on in the brain as we've gleaned from our functional brain imaging work. And what we found using fMRI before and after the treatment was something quite intriguing. And it was intriguing because it didn't fit what we see when we look at the brain during a psychedelic experience. So here we're doing the imaging one day after the treatment. People have returned to sobriety. They're often feeling quite well. And so we rescan them. And here we saw that brain networks that ordinarily disintegrate, at least relatively speaking and transiently, under the psychedelics seem to reintegrate afterwards. And we actually saw a slight spatial expansion in the default mode network after the treatment. And this effect correlated with the longer-term efficacy of the treatment. So those who were maintaining response at 5 weeks, for example, showed this increase in default mode network integrity to the largest extent. So it seemed to be related to the action, the therapeutic action of the intervention. Then looking at other treatments for depression, it was interesting to find that similar effects have been found with electroconvulsive therapy. So this idea of a kind of reset or recalibration in the brain with certain treatments is one that you could use in the context of psychedelic therapy, at least to an extent. We also saw drops in blood flow in the amygdala, which kind of made sense in terms of the context of the literature on depression, finding hyperperfusion, too much perfusion, too much blood flow in the amygdala, for example, during a depression. We also saw that the amygdala, contrary to what you see with SSRIs, where there's a kind of blunting of the amygdala response to emotion, we saw that it became hyper-responsive to emotional stimuli after the treatment. And again, this effect, although it's in the opposite direction of what you see with SSRIs, it correlated with the efficacy of the treatment. So again, those who were maintaining response into 5 weeks was showing this increase in responsiveness of the amygdala to emotional stimuli. So one way we've characterized this is that people, instead of there being a kind of moderation of stress and perhaps a moderation of emotion with other treatments for depression, other drug treatments for depression like SSRIs, with psilocybin there's more of a reconnection perhaps to one's emotion after the treatment. Now, in terms of understanding how this treatment can be effective, I've recently got onto this topic of quantum change. And I know quantum's a term that's popular at this conference. I don't really mean it in any sort of physics sense. It's more just the idea that there's a sudden change. And this is a principle that we accept quite readily in the context of trauma, for example, that with an isolated extreme experience, in the case of trauma and negative experience, it can have lasting consequences. So couldn't the same be possible with an intense experience that's positive? Could this not have lasting, enduring changes? This idea fits nicely with some of the findings with psilocybin and other psychedelics when given to humans, that they can change people in quite an enduring way. So, for example, the team in Johns Hopkins has found increases in well-being over a year on after a single administration of psilocybin, increases in the personality trait openness as well, and this is something that we've replicated in other teams now as well. So this key question of what mediates that, we found recently, as others have, that the quality of the experience that people have under a psychedelic seems to be key. and more specifically, that if people have what you might call a peak experience, if we're borrowing from Abraham Maslow, or in other vocabularies people call a mystical experience or a spiritual experience, then they're more likely to have the long-term therapeutic, positive therapeutic outcomes. What is the peak experience? It was described as follows by Abraham Maslow. It's the feeling of being at one with the world, a sense of unity, intense happiness and well-being, wonder and awe. So Diana Fosher, who's written about quantum change, has developed this accelerated experiential dynamic therapy where she describes using therapy in a particular way to put focus on emotion rather than cognition, with the aim of in repeated therapy sessions to generate these alternating waves of experience, and then after the therapy sessions, reflection. So these themes, I think, are important when we're thinking about how this treatment works, and also just, instead of being too focused on psychedelics, thinking about other states. Something that I'd like to get a better handle on is the relationship between this principle of the entropic brain and arguably the dominant model of how the brain works, if you want. At the moment, the so-called Bayesian brain model, that the brain is a prediction machine that sort of stumbles its way through experience and updates its predictions about the way the world is in order to more accurately navigate the world. And this idea that if that's all the brain did, that it just learned from surprise and so tried to maximize its confidence about the world, perhaps an individual might just be so assured that they don't really learn anything new, that they in not making mistake anymore, they never generate anything new. So Carl Friston, who's written a lot about this, has recently put more of a focus on how curiosity and exploration can allow for further development in learning. So this idea of there being an opportunity in an entropic hot state for updating one's priors about the way the world is. Just coming towards the last couple of slides now, this link between, as I said, coming full circle now, this link between the function of serotonin receptor stimulation and it relaxing assumptions, how this may play a role in development, how it may play a role in psychedelic therapy, the evidence that's supporting that, the importance of context, speaking to this principle of the 2A receptor being so crucially important in terms of sensitivity to environment, how increases in brain criticality, something that I hope Selenatozoy will talk to you about in the context of psychedelics, increasing not just brain entropy, but also properties of criticality during the psychedelic experience. One key property of criticality is that a system is maximally sensitive to perturbation. So that would fit with the idea of the brain and the mind being more sensitive to the environment under psychedelics, both internal environment and also the outside environment. In the interest of time, I'll probably skip over this slide, but another principle of if you perhaps jiggle up the brain, then there'll be increased opportunity for insight and new learning, and that might come within, and perhaps some of the dangers of supposed insight under psychedelics, whether it's always veridical, it's a key question. So yes, just ending now in terms of the developmental function of 2A receptors, that they may tune learning by relaxing priors. Alison Gopnik has shown that infants are particularly adept learners in the sense that they don't come to the world with such firm assumptions, and so can sometimes perform better on certain paradigms than adults. And also, if we think about this 2A receptor in terms of the evolution of the brain, that perhaps it's played a crucial role. And I don't mean that it's played a role, as, for example, Dennis McKenna proposed, that people took psychedelics that altered consciousness in a certain way that it conferred some behavioral advantages, some evolutionary advantages. I would offer a more sober explanation, perhaps, that serotonin may have done it, and serotonin is released during states of stress. I showed that stress also promotes, more specifically, serotonin 2A receptor functioning, and so maybe this particular receptor that psychedelics modulate has played a crucial role in the evolution of human consciousness. With that, I'd just like to thank you for your attention. There we go.
Thank you, Robin, for that fascinating and enlightening talk. It gives me great pleasure to welcome up our next speaker, Dr. Jimo Borjigin, faculty member at the University of Michigan, who has expertise in circadian biology, but has recently turned her attention to consciousness. She's done a lot of great work on the near-death experience, and I look forward to hearing her work on DMT.
Okay. Well, good afternoon. I'm really pleased to be here. This is actually the second time I'm here. So last time, for those of you who are here, if you still remember what I talked about, which is physiological basis of a near-death consciousness, I know it's a highly controversial topic, but some of you, for those who were here, you probably were thinking that I'm trying to link the near-death experiences with DMT. I know that if you go search the internet, you're going to find lots of stories and people's testimonials who propose that the two are linked. If you're on my thoughts, I'm probably going to disappoint you because I have no data showing you for that aspect. But I am really excited about the opportunity today because everything I'll be showing you, it's brand new, have not published yet. So George is pretty psychic knowing that I have something to say about DMT. Having me here. Excited because I think finally we may have a concrete molecule to study in terms of molecular basis of consciousness. Okay, so the title of my talk is going to be Evidence Supporting DMT as an Endogenous Neurotransmitter of the Brain. Okay, so my connection with DMT is actually another accidental discovery, just like my connection with the near-death experiences. It's all from accidental discovery of my experiments in the lab. And over here, because I've been teaching graduate students about circadian rhythms, one day I was just searching the internet, trying to find some really fun, entertaining slides for my students to understand circadian biology and pineal biology. So what I discovered is something fascinating. And on the YouTube, I discovered that Dr. Rick Strassman, in talking about the DMT and talking about the pineal gland, I was fascinated because I thought, wait, I've been studying the pineal gland since 1994. I've been to a lot of pineal Gordon conferences, scientific meetings. I never really heard anything about DMT before. I thought, where is this from? So I got very disappointed in myself. I said, please tell me where you got this reference from. So I sent him an email. This is 2011, asking where is the reference for that claim that a DMT is produced by the pineal gland. Okay. Dr. Strassman has been extremely kind, encouraging ever since. So we've been collaborating ever since this email was sent. Okay, so then I thought, well, the history response is that that was just my best guess. I thought, oh, wow, best guess. Let me see how he guessed it. So this is a chemical listed. Oh, over here, you can see that everything in the pineal gland seems to be coming from the amino acid tryptophan. Tryptophan is everywhere. It's in our circulation. If you eat something tryptophan-rich, you're going to have tryptophan all over your body. So if it lands in the cells that has enzymes that convert tryptophan, then you're going to have fascinating molecules. So first of all, you see tryptophan is, I don't know whether you can show this one. Okay, so tryptophan here is converted to serotonin in the pineal gland. In fact, serotonin has the highest concentration in the pineal gland anywhere in the body. And the serotonin is then converted to melatonin. Melatonin is the molecule that I've been working on for decades. And in fact, my postdoctoral career began with the cloning of the enzyme that makes melatonin in the rat pineal gland. That was surprisingly, it was really and opened up my career. But if you look at it, the tryptophan is then also converted to tryptamine in the pineal gland. And because of the enzyme aromatic amino acid decarboxylase, AADC. AADC is the essential enzyme for serotonin production. Therefore, it's there in large quantity in the pineal gland. So what Dr. Strassman made is this leap from the tryptamine to DMT in the pineal gland. I thought, well, at the time, I don't think anything is known about the existence of the enzyme in the pineal gland there, but I thought, it's possible. Let's collaborate. So I emailed Dr. Strassman again. I said, why don't we work together to confirm this is really the case? Okay, the result of that is the publication of our paper published in not so high-profile journal, but actually surprisingly attracted lots of attention. In fact, it was the #1 ranked paper in that journal. I was rather surprised and actually honored to be in a research article that attracted such social attention. And that's when I really realized that DMT is a fascinating molecule for a lot of people. So now the question is DMT synthetic enzymes are present in the brain? Yes. In fact, if you dig into the old literature, Axelrod in 1961 discovered enzymatic activity converting tryptamine to DMT using rabbit lung extracts. So we knew that a rabbit lung actually produces, has the enzyme activity to make DMT. And subsequently, Morgan and Mendel, also in the 60s, late 60s, discovered using human brain extracts or sheep brain extracts, the enzyme activity is also present in the brain. So this is, and subsequently, in 1972, again in Axelrod's group, they discovered that rat brain has the same enzyme activity. Therefore, so the brain has the enzymatic activity to convert tryptamine to DMT is pretty solid, this story. Now the question is, how do you study that? Just knowing the enzyme activity is very unsatisfying. You want to know the enzyme, you want to clone it, you want to study the DNA, you want to study the translation, post-translation modification of the enzyme, so you can get molecular understanding of the DMT. Okay, so fortunately, Michael Thompson in 1998 published a paper about a cloning of the INMT. I think I probably wanted to tell you the abbreviation, so that I'm going to use a lot. The AADC stands for aromatic amino acid decarboxylase, the first enzyme for DMT synthesis. Second enzyme is INMT, stands for indoethylamine N-methyltransferase. So INMT is the second enzyme, which is one we're going to talk about a lot. Okay, so if you look at the northern blot analysis, this is a method to detect the level of RNA expression in brain tissue, in various tissues, in fact. So you can see that lung has the highest concentration of this RNA species. It's around 2.6 I believe that's about the size of that species that is very highly concentrated in the lung tissue. And if you look at the brain, the level of expression is very, very low. Okay, you have to expose the blood, which is radioactively labeled, for more than a month to see anything there. Okay, so since then, since 1998, and subsequently the same gene was cloned from human tissue in 1999 by the same group of people, Thompson. It's found in the human spinal cord, so this is an RNA at the time still, and subsequently it's discovered to be in the primate pineal gland, which fits very nicely and must be satisfying for Dr. Strassman to learn about this. So this is published in 2011. In 2012, a study discovered that this is also, the enzyme is also present in mouse spinal cord. We still don't know why it's there, but it's something that we wanted to study. So after we published a 2013 paper showing that DMT is secreted from the rat pineal gland, this is the rat pineal gland, actually in living, free-running rats. So we thought we got to make sure the DMT production in the pineal is actually locally made. It wasn't something made in the lung or liver or somewhere else that somehow just carried into the pineal gland and collected by these dialysates. So we wanted to clone, we wanted to identify the genes and proteins in the pineal gland. Okay, this is then so this 2014. Okay, this is the year that my student, John Dean, joined my lab as a master's student. So this is in situ hybridization showing the expression of INMT message, okay, in the rat pineal gland. You can see this very high level of expression here. And again, Dr. Strassman is very happy that his hypothesis was confirmed finally. Now we have a concrete evidence. We know that AADC is highly abundant in the pineal gland. Therefore, there's both enzymes are there. It's highly likely the DMT is made within the pineal gland of the rat. But in addition to the pineal gland, we found DMT synthetic enzyme, the INMT, in cortical neurons. Until the Michael Thompson's experiment, which is a northern blot study, is showing that very tiny amount of RNA is found in the rabbit cortex, or rabbit brain, actually. We don't know where exactly they got the tissues from. And this is really the first study showing that DMT synthetic enzyme is expressed in neocortical neurons. Okay, so this showing that you can see that some neurons have very high level of RNA expression, some have very little or maybe none. And this is also true in human frontal cortex. Okay, so we were really excited about this, but we thought, okay, do we know what in the cortical neurons, the both enzymes for DMT synthesis are located? Okay, if they're not located together, it's probably not likely that in those INMT expressing neurons that DMT is actually going to be made. So this is actually the idea that we had to confirm. That is, since DMT synthesis from tryptophan requires both AADC and INMT, we needed to confirm both enzymes are in the same cells. So not too long ago, this is October last year, I searched through the internet. This time it's Allen Brain Institute. They have a whole panel of in situ hybridization results from mouse brain, looking at probes or known genes or unknown genes, a whole long battery of genes that are analyzed. So if you look at the genes, can you dim the light a little bit? I don't know whether you can see the RNA expression there. So what I was trying to show is that on the top here, this is called in situ deoxygenated in situ hybridization. So the final reaction is using enzyme substrate. So you can see the cells that has high level of expression would appear to be darker bluish, such as these shown here. And in order to sort the cells that has higher level expression from those simply just hematoxylin positive cells, there is a so-called expression subtraction that is actually a procedure conducted by the Allen Brain Institute to show you where the genes actually expressed at the appreciable levels. Okay, so if you can't see those cells, just take my words for it. That is INMT on the top, the ADC on the top here. It's expressed in high levels of choroid plexus up here. It's also appreciable levels in the cortex, both in the frontal cortex, occipital cortex, as well as in the cerebellum. In addition, it's found in the hippocampus. Okay, so this lighting is a little bit hard to see. And the lower panels over here, panel B, shows you the corresponding expression of INMT in similar brain sections. These are not from the same animal, so it's not really adjacent section. But you can see that, for instance, INMT expression over here is also very high in the choroid plexus. It's identifiable in both frontal cortex as well as occipital cortex. It's also found in discrete cells in the cerebellum. In addition, it's in the hippocampus. Okay, this seems like the two are in the parallel fashion. That is and we didn't know before because we never conducted such a thorough in situ hybridization analysis of these two genes. So we said, okay, John, you need to do this study. We need to show that the two genes are found in the exact same neurons. This is what he did for his weekend hours after 5 p.m. Anyway, so everything worked out perfectly well, and which is really great. I was very happy to see these results. What you see here is that both of these in situ hybridization used a color substrate in the end. So for INMT expressing areas, you can see there are little bluish dots. See that INMT is blue, so the bluish dots dotted all the neurons most of these neurons in the cortex right here. Okay, the AADC expressing cells showing the red or pinkish staining. You can see that over here, almost every cell that is positive for INMT in bluish stainings, also positive for AADC. This suggests in the cortex, the two genes are co-localized, they're found in the same neurons. Okay, this is true almost wherever we looked at, okay, except in the brainstem. We're not going to show you. We have immunostaining data demonstrating that. What you see is that, for instance, over here in the choroid plexus, you can definitely tell these neurons are double positive for both INMT and AADC. Over here in the hippocampus, level expression is lower, but you can still see whichever cell you focus on, you would have both the blue dots as well as pink dots, suggesting wherever really AADC is found or INMT is also found in the brain, okay, at least outside this brainstem. So that means that these neurons are showing both expressions. These can be called DMT neurons or D neurons or psychedelic neurons if you want to. So we're so happy about these results. The previous double in situ was only obtained really recently, but we had several other pieces of information, so we submitted to the several journals and rejected one after the other. That's one thing that I learned is that in the scientific community, especially among neuroscientists, there's a fierce resistance to the idea of DMT could be endogenously made. That somehow there's actually our brain synthesize the DMT. It's either that or it's too controversial that we have to be super compulsive in showing them every single possible controls we could do before they could give us the credit. So this is a response we got from one of the reviewers. So this one here says, cortical neurons do not contain any proteins that could theoretically transport DMT. Okay? Therefore, the conclusion is that the absence of potential mechanism that would allow cortical neurons to release DMT that provides evidence that expression of INMT in neurons has nothing to do with the DMT production. I said, wait a minute, is that true? Okay, so I gotta go back to the textbook to find out where the transporters are expressed. So just until, before we can all be on the same page, understand what the reviewers are saying, is that I wanted to just go refresh our memory about what it is for a chemical to be neurotransmitter, what does need to be, what are the other mechanisms that the cells need to have in order to call something a neurotransmitter. Okay, so it requires two distinct transporters. Okay, so neurotransmitters, once they're made, they need to be packaged into synaptic vesicles in the synaptic terminals. Okay, so that function is served by vesicular neurotransmitter transporters. Okay, so every single neurotransmitters we know of possess one such transporter. Sorry. Okay, for instance, what I'm showing you here is the 4 types of neurotransmitters. Up here, that can go up. Okay. Over here, this is acetylcholine, cholinergic synapse. And down here is, if I can see this, a glutamate. And then over here is a GABA. So all of these have really well documented transporters for their vesicle packaging and storage. Okay. So let me just, so this is the vesicular transporters. In addition to the vesicular transporters, you need to have plasma membrane transporter. And these transporters serve to recycle the released neurotransmitter in the synaptic cleft so that they can be used for future release. So let's focus on the transporters for serotonin transport. Okay, serotonin belongs to this group called monoamines, okay, that includes dopamine and the norepinephrine. So all 3 transporters are packaged, once synthesized in the synaptic terminals, packaged into synaptic vesicles by a protein called vesicular monoamine transporter 2. Immediately you wonder, what is transporter 1 then? Okay, so I'll tell you that a little bit later. And so for the plasma membrane transporters, there are actually 3. This is the second one I should go to. So there are 3 plasma membrane transporters for monoamines, 1 for each of the distinct neurotransmitters. For instance, serotonin is transported or recycled back by the serotonin transporter, the SERT, whereas norepinephrine is transported by the norepinephrine transporter, or the NET. The dopamine has a dopamine transporter called the DAT. So all 3 are located in the plasma membrane to recycle the released neurotransmitter. Okay, so that means if DMT is a neurotransmitter, DMT has to have some transporters like these to be responsible for packaging of DMT in the synaptic vesicles and for recycling once DMT is released in synaptic cleft back into the presynaptic terminal. Okay, so I said, well, I know there's evidence. Somebody did experiments to show that DMT can be transported by the VMAT2 as well as the SERT. Okay, in fact, this data is provided by Dr. Cozy in 2009. Even before that, the synaptic vesicles isolated by researchers were able to package DMT without knowing what are the transporters that are doing the activity. So these data suggested that DMT can be transported into the vesicles. So in fact, this study is really highly cited. Anytime anybody wants to talk about DMT as an endogenous hallucinogen or neurotransmitter, these papers are cited routinely. Then I thought, wait, aren't they expressed in the cortex? I'm sure it's there. If you look hard enough, it must be there. So I actually searched through the Allen Brain Institute, the mouse atlas, didn't find any. So it's true, the reviewer was correct. These monoamine neurons, or the associated transporters, the enzymes, are all found in the brain stem. Okay? So this one actually demonstrates the serotonergic neurons located in the raphe nuclei. It's actually several clusters of neurons in the brain stem, and showing the projections of exons. You can see the serotonergic neurons project to everywhere. Can't find any place that it's not projecting to. It's a highly important, and I think Robin already told you a lot about serotonergic systems that I'm not going to repeat here. So the thing is this, but this one over here, so the serotonergic cell body is not found in the cortex. That means that VMAT2 is also not in the cortex. So what could it be? So then we had to build a model. It says there is a DMT-nergic synapse in the brain. Okay, there got to be a transporter, vesicular transporters transport DMT into the vesicles. There has to be a plasma membrane transporter recycles DMT once it's released in the synaptic cleft. So these two transporters have to be discovered, have to be identified before we can call DMT a putative neurotransmitter. So since the vesicular monoamine transporter 2 is called a 2 for a reason, so I said, what about a transporter 1? Where is it? Where is it found? So I went through literature, spent a week reading hundreds of articles, and I learned a lot. And this is actually, this slide lists some of the things I learned during that October week. So I found that there are actually 2 vesicular monoamine transporters, VMAT1 and VMAT2. VMAT2 is the one who's responsible for vesicular packaging of all of the monoamines, including serotonin, dopamine, and norepinephrine. Whereas the VMAT1 was initially identified in the adrenal gland in the peripheral bodies. There's very little expression in the brain. But later on, people found that a VMAT1 is actually diffusely expressed at lower level all over the brain. Okay, no one knows what it does. And VMAT1 is expressed in lower, it is expressed in the cortex, but the level is very low, but the VMAT2 is very high. And interestingly, there are actually lots of knockout animals and studies being done for these transporters. In VMAT2 knockout mice, if you genetically knock out the VMAT2, the brain levels of serotonin and other monoamines are severely depleted, suggesting in vivo, in living animals, the VMAT2 is responsible for packaging of all these monoamine neurotransmitters. So in these knockout animals, even though VMAT1 is still there, it doesn't do the job. That is, in vivo, VMAT1 does not transport serotonin or other monoamines. On the other hand, there's the VMAT1 knockout animals. Okay, so VMAT1 deficient animals has no reported deficits and no monoamine neurotransmitters. Okay, so what does that mean? That means the ligand for VMAT1 is unknown. So VMAT1 is kind of like orphan transporter in need of neurotransmitter to transport. So we then examined the expression of the VMAT1 in the mouse brain, shows yes, they're actually parallel. We mean that I did the study, and we said, okay, John, we got to do this study again. So he did double in situ and showing that, in fact, INMT and VMAT1 are perfectly co-localized in the cortex, in the choroid plexus, as well as in the hippocampal neurons. So now we know VMAT1 could very well be the vesicular DMT transporter. So what is a plasma membrane transporter? So plasma membrane monoamine transporter is widely expressed in the brain, including cerebral cortex, hippocampus, and choroid plexus. And it's known to transport serotonin with a high efficiency. It's actually higher than any other transporter. It's known. And there's no reported rise of extracellular monoamine concentrations in the knockout animals, suggesting in vivo PMAT1 actually transports something else, yet unknown. Okay? So we said, okay, we got to do this study also. Let's do double in situ. So again, doing the in situ search for the available data showing their parallel expressions. But when we do the double in situ, again, these 2 genes are co-localized beautifully. You can see that in the cerebral cortex, high-level expression found in wherever the INMT is. PMAT is also there. This is true for the choroid as well as the hippocampus. So that means that PMAT may be the plasma membrane DMT transporter. So what that means is that we now have a model for DMT as a neurotransmitter. So we found both the vesicular monoamine transporter 1 as well as plasma membrane monoamine transporter to support DMT's role as an endogenous neurotransmitter. So what is the implication here? I think the implication is that a DMT is endogenous synthesized and released from the cortical neurons, may function as a non-classical monoamine neurotransmitter and may play a role in cognitive functions. And dysfunctional DMT transport may be associated with human psychiatric disorders that are currently known to be associated with the mutations in patients with the VMAT1 and the PMAT genes. Okay, so this is where I think is a really going to be exciting. We can talk about more of this implication in the discussion sessions, but I'd like to thank the people who are really important for this work. First of all, the Dr. Strassman and Dr. Barker were the ones who helped me start this project. It's extremely supportive and very helpful. Of course, George, you all know, has been a strong supporter of this particular project. And DMT, I still remember the first time when I talked about DMT to him, his eyes just lit up. And I said, DMT is amazing. And John, he spent a year in my lab, got a master's degree, who was the one who did all the in situ studies, and with lots of help from Dr. Michael Wang's group. So we're going to ask questions for the after the next talk. Thank you.
Thank you very much, Jimo, for that exciting lecture. I also want to commend you for being so open and sharing these unpublished data with us. Very exciting. It's a great pleasure to welcome up the third and final speaker in this session, Dr. Anil Seth, who is the director of the Center for Consciousness Science, the Sackler Center for Consciousness Science at the University of Sussex. He's also the editor-in-chief of the Neuroscience of Consciousness, which is published through Oxford University Press, and a renowned expert in this field. So please welcome him to the stage.
Well, George is very kind, actually, in saying that I'm an expert in this field. This isn't really true at all. I came here for the first time 2 years ago, and I hadn't explicitly done any research on psychedelics, and it was only thanks to really meeting both Jimo and Robin, actually, and spending quite a substantial part of the conference by the pool talking about his research in psychedelics that we realized there was a lot that we could do together. So I really want to thank Robin for generously involving me in this area of exciting research. So I'm going to tell you in the next 30 minutes about some studies that we have been doing in this area, both on neuroimaging of the brain under psychedelics, and also some other ways we've been trying to get at simulating or generating psychedelic-like experiences in the lab, because my basic interest here is in what the mechanisms of conscious perception are generally. And I think psychedelic states are a uniquely powerful angle to try to understand this fundamental question in consciousness science. So why study them? Well, there's what I just said. It's a brilliant window into the mechanisms that shape, that give rise to our conscious experiences of the self and the world generally. There are also very important clinical motivations. We heard from Robin about these. And there's increasing work in this area. You've seen a lot of it already today, but still, I think it's fair to say that the effects of psychedelic compounds globally on brain dynamics are still not very well understood. This is just an image from Bicycle Day 1943, the renowned day when Albert Hofmann cycled home, having first experimented on himself with these sorts of things. And I just wanted to start by reminding ourselves, since we're probably not going to just down some of this stuff right now, what the effects are on our phenomenal experiences. And I love this quote from Hofmann. Little by little, I could begin to enjoy the unprecedented colors and play of shapes that persisted behind my closed eyes. Kaleidoscopic, fantastic images surged in on me, alternating, variegated, opening, and then closing themselves in circles and spirals, exploding in colored fountains, rearranging and hybridizing themselves in a constant flux. And this really emphasizes not so much the dissolution of the ego aspect, but really the incredibly vivid perceptual effects of these compounds. So what's going on with these effects? Well, I want to tell you about a few different things today. I'll start with some of our research directly following some of Robin's ideas about entropy, but getting a bit more technical about how we measure entropy in the brain in these conditions. And then some new results that I haven't described anywhere before yet, we're still working on these, which show good reason to think that the psychedelic brain has decreased information flow between regions of the brain quite generally. And this, I think, has some interesting implications for studying connectivity in the brain more generally, not just in the psychedelic state. And then I'll move on to some of the other stuff where we're not studying pharmacological manipulations, but trying to induce hallucinatory experiences by other means. Strobe lighting is a surprisingly powerful method for doing this. And then we've also been doing some rather fun stuff with virtual reality and deep neural networks to simulate other aspects of perceptual phenomenology in these states. So let me start with the first bit, signal diversity. This is Robin's paper. It's not the newest one, but it's one of the first articulations of this idea that the psychedelic state in the brain is reflected by increased entropy, a sort of broader repertoire of possible states the brain could be in. Now, this, I think, some interesting relations between this idea and some other theories about the necessary and sufficient mechanisms of consciousness that have reached a lot of prominence in recent years, one in particular being Giulio Tononi's integrated information theory, which is sort of, it's a different view on the role of entropy, because this theory of Tononi's emphasizes both the range of possible states the system could be in, but also their behavior as a unified entity. So mathematically it gets quite complex, but I think they're definitely related, and we begin to get some empirical handle on this relation through the study of psychedelics. So how do you measure signal diversity? How do you measure entropy in the brain? How do you measure the repertoire of states? Well, what you'll see a lot in this talk are measures of what we call Lempel-Ziv complexity. I'll explain a little bit how it works in a second. But let's just go back to entropy. Entropy, technically, is a measure of the possible states a system could be in. It's the uncertainty about what state a system could be in given the possible states that are on offer. It's very difficult to measure given some data. You need a lot of data to get reliable measures of entropy. Lempel-Ziv complexity is sort of an approximation to entropy which works well for the kinds of data that we generally have available. It's basically a compression algorithm. Every time you take a photo and send it, it will be compressed into probably a JPEG format or something like that. This uses exactly the same algorithm to come up with what we call the minimum description length for some data. And to try to put it very simply, imagine you've got just a string of ones and zeros. Now, this could be a digital photo, or it could be a measure of the brain in any particular state that's sort of binarized into a string of ones and zeros. What you want to do is find the shortest description of that string of ones and zeros that is capable of regenerating the whole string. If you have a string that's just a load of ones, it's actually very simple. You just say 1 n times. The minimum description length can be very short. There's no uncertainty in a string that's all ones or all zeros. If it's completely random, there's no way to compress it. You have to re-specify every 1 and 0. But if it's somewhere in the middle, you'll be able to compress it to some extent. And Lempel-Ziv is a measure of that extent. So basically, you look for repeated patterns of different lengths, and then you ask what's the size of the dictionary of these different patterns that you call upon to regenerate the sequence. But basically, you can just think of the higher this measure, the greater the diversity of states represented in the sequence, roughly the more entropy there is. The nice thing about this, it's very applicable to spontaneous electrophysiological data of the sort that you get in EEG and other measures. And one thing we have to be very careful of, and I will come back to this, is that in any measure of brain dynamics, lots of things change. It's very difficult to just isolate one thing that changes. So when we make claims that the diversity or entropy of something has changed, we have to try to say that this is something that's over and above what you would expect from other changes that we already know about. So we know, and you've seen this in Robin's, you see in talk that you see strong changes in spectral power. For instance, alpha power goes down a lot. And we have to try to show that a more complex measure, like signal diversity, is not just a trivial reflection of something more simple. And that's, I think, generally true in neuroscience too. So there's some background for doing this. We've been working with this measure for a number of years in different contexts. This is data from anesthesia, and what we see here is that this is just EEG, surface EEG data. What we see here is that complexity goes down as anesthesia deepens, so there's a loss of signal diversity, a reduction in entropy under anesthesia. The same is true for different stages of sleep. This is data we analyzed with colleagues in Italy, taken from intracranial EEG. So this is when you've got electrodes stuck deep into your brain, and divided here into 4 areas. On the left, you just see different part where the electrodes ended up. These are all done for clinical reasons, so they're all over the place. And what we see, again, is that complexity, or entropy, or diversity goes down, especially in the non-REM stages of sleep when people are generally not dreaming or not having rich dream content. And in REM sleep, where rich dream reports are more common, levels of entropy are pretty much indistinguishable from the waking state. So that's also quite nice. Now, this is where the conversation with Robin came, and we thought, well, let's do the same thing then with the data that he has on the psychedelic brain. And so we used MEG data that he'd collected with Suresh Muthukumaraswamy at Cardiff. This is an MEG machine. MEG is a lovely technology, measures the magnetic field fluctuations that are caused by populations of neurons firing together. It's very high time resolution, like EEG, but it's better spatial resolution than EEG. The reason being the magnetic fields that we detect with MEG are not distorted by the skull and the scalp, which mess around with the electric fields in EEG. Let's just give you an idea of what we're talking about. I don't know who any of these people are in this photo at all. Let's just say that. So this is just some data from Suresh's and Robin's original paper, where, of course, we want to know what's the subjective effects. And so this is just using something called the Altered States Questionnaire. Many of you will probably know it. It's a way of just formalizing the phenomenological effects of a manipulation. And here you can see very high ratings for the three of these psychedelics, ketamine, psilocybin, LSD. You see high ratings for strange experiences, vivid experiences, distortions of time, and whatnot. And I'll show you how these questionnaire results can be also applied to some of the other ways of inducing hallucinations I'll be talking about. So if we look at power first, what happens to just the spectral power, the strength of the signal in different frequency bands in these conditions? Well, what you see here in these plots are the 3 different drugs, the number of subjects in each condition. And again, this is all data that was collected by Robin and Suresh, not by us. And when it's blue, what that means is power in that area is going down. And when it's red, it means power in that particular area is going up. And what you see in the top row, each little brain is a different frequency band. The top row is generally the lower frequencies, up to about 30 hertz, including alpha at about 10 hertz. And the bottom row is either the higher frequencies, the gamma frequencies, or the broadband, which is all frequencies. And so what you can see here is that the lower frequencies go down, and the higher frequencies go up. We have to be very careful here, because high frequencies like gamma oscillations are very affected by artifacts. And if you move your eyes a lot, or you've got fine muscle tremors, or you smile a lot, you can see elevated results in the gamma frequency. So I'm not sure whether these are actually to be trusted, but the decrease in power in the lower frequencies is a very reliable effect. If we actually look at relative power rather than absolute power, though I quite like the idea of absolute power, if we look at relative power, you see this is the sort of fraction of, if you factor out the overall change in power and look at how it's distributed among different frequency bands, it's mainly in the lower frequencies. So I think that's where the interesting stuff is actually going on here. So this is stuff we already know. But if we now look at the signal diversity, these measures of complexity, levels of complexity, what we see is this. So these are similar plots. Here, what we're showing in red are those areas of the brain. Now, this is MEG. These are source localized. So we've divided the brain up into 90 different regions. And where it's red are where the entropy, the signal diversity, is higher in the drug state than in the placebo state, significantly higher according to a t-test between the two conditions. And also importantly, these are normalized for changes in power. So we know the changes here are above and beyond that which you would expect just through the changes in power. And quite clearly, and on the bottom you see per participant, pretty much across the board, entropy goes up. There is higher diversity and entropy in the brain on the psychedelic compounds than not. And this is really, I thought, quite striking, not only because it's so robust, which is always something very nice to see, But because all the other measures of complexity that I'm aware of, and that certainly we've been involved with, always show reduction from baseline. You have your waking state baseline, everything goes down. This is the first example that we'd found or described, and I think it still is, of these measures going up. Of course, this meant that, this then got written up by the various newspapers as evidence for a higher state of consciousness, which was like, why do we need evidence for that? We know that anyway. But they didn't quite get what we were actually doing, and evidence for increased signal diversity in the psychedelic state didn't make such good headlines. The Daily Mail, for those of you who know that terrible newspaper, had to accompany it by a big sidebar on the dangers of LSD. I dropped this slide in just now because there's ongoing work, and I think I'm not particularly involved in this now, but my student, Michael Shartner, who did these analyses, has been working with some of Robin's people as well to look at the DMT data, and we see pretty much the same thing here. It's quite a complicated graph because it's still preliminary stages, but basically the lines that go up and then down, that's the signal diversity, and the lines that sort of go up are the power. So you can see in that where that arrow is, that's the period of the main effect of DMT, and you can see both this dip in power and the rise in levels of complexity and signal diversity. So we see the same thing in DMT as well. If we just superimpose that graph that Robin had, it follows very nicely. You can see the intensity of the experience goes up, comes down, and the complexity, with a slight delay, comes up. So this is still very much ongoing work. So the next thing we did was, rather than just looking at entropy, we wanted to look at information flow. So how do different brain areas change the way in which they communicate or speak to other areas in these states? And there are different ways you can think about connectivity in the brain. There's structural connectivity, what's connected to what by a bunch of axons. What's the wiring diagram? Then there's functional or dynamic connectivity. How do different regions change their activity together, dependently or independently? And then there's what some people have called effective connectivity, which is a kind of mechanistic causality. How does one brain region influence the activity of another brain region? These are all distinct readings of connectivity. And the one in the middle, functional connectivity, importantly, can be split into two. You can have undirected functional connectivity. This is mostly what most of the people do most of the time. This is looking at things like correlation, coherence, synchrony, mutual information. It's just how do signals change together? You've got your X1 and X2 there. Imagine they're just two parts of the brain. Do they go up and down together? That's undirected functional connectivity. But you also have directed functional connectivity, and this is key. This is something completely independently we've been working on in my lab for about, well, me anyway, for about 15 years. And it's a way of saying how is information flowing between two variables. So it's like you put an arrow on your connectivity here. Is X1 pointing to arrow 2 or X2 pointing to arrow 1? What does that mean? Well, it's typically measured by this quantity called Granger causality, and I'll explain very briefly what that is, because that's what we're going to see in the results. So Granger causality says, and causality is really the wrong word for it, it should be predictability or something, says that information flows from X1 to X2 if knowing about the past of X1 helps you predict the future of X2 better than just knowing the past of X2. So this operationalizes two ideas about what it means for information to flow or something to cause something. Something has to come before the thing it's causing, and it has to help you predict that thing's behavior. And it has to help you predict that thing's behavior better than you could otherwise do. So we measure this quite simply by building simple mathematical models that, say, try to predict the evolution of these two variables using all the information available, in this case both X1 and X2, and then we'll drop the potential cause and we'll see if our ability to predict X2, the potential effect, is affected. If it stays the same, then X1 wasn't really contributing any information. If it goes down, it means X1 was helping you predict X2 better than, and it's helping it in a way that wasn't already available by knowing X2. So X1 causes X2, there's information flowing from X1 to X2. And we can put a number to that, and we can extend it to many different time series. And it's a very different measure to correlation and coherence, talking about information flow. There's a very long and complicated mathematical paper we wrote that if you want to do this at home, calculate Granger causality, you can do it this way. I won't go into the things there. If you want to think about it more simply, I love this cartoon from one of this online magazine's ex-seed. I used to think that correlation implied causation. Then I took a statistics class, and now I don't. Sounds like the class helped. Well, maybe. That says it all. Now, the results. If we look now at correlation change in the brain on psychedelics, again, red is more. We see increased correlation. The different parts of the brain are changing in together more tightly in each of these conditions. But if we look at Granger causality, so this measure of information flow, it goes completely the other way. Again, very robustly across all drugs, pretty much all brain regions, we see a decrease. So here, what I'm plotting for each region is the mean connectivity of that region with all other regions. Not specific one region to another, but the mean connectivity globally across the brain with each brain region by turn as the source. And so that's why you have inbound and outbound at the bottom as well, because it goes in both directions. But whichever way you go, it's less. And this is also true by frequency band. If we look at coherence, which is undirected, it goes up. If we look at Granger causality, either inbound or outbound, it goes down. So there is less information flow in the brain, but you can only see that if you use these explicit measures of information flow. They go in different ways. This is interesting because we kind of intuitively assume that they would probably do the same thing, these directed and undirected measures. Certainly, I was not aware of any other situation which showed empirically them moving in different directions. And my colleague Lionel Barnett, who's a mathematician in my group, was also surprised and did a lot of mathematical modeling to show that this is in fact not surprising, which is good. And he actually solved for a very simple model what correlation and Granger causality would look like as you sweep through the whole parameter space. And what you see here, basically, these two graphs look different. What that means is that even for a simple model, Granger causality and correlation don't go together. You can find parts of the parameter space where they do, parts where they don't. And so this is, I think, quite a general result that whatever part of neuroscience we're looking at, we need to measure both of these things in order to make any claims about the dynamics of the brain in these conditions. Not enough just to look at correlation, because information flow can do very different things. So to summarize this bit, increased signal diversity in the brain following ingestion of these psychedelic compounds, which are driven partially but not totally by changes in power. I think this is also, it's interesting that these measures of Lempel-Ziv, as I introduced, they're generally applied to things like sleep and anesthesia. So big gross changes in conscious level, you lose consciousness or not. Here we've shown a change applied to conscious content. You know, people are still awake when they're having these experiences. And so we're finding an empirical bridge between these two areas of consciousness science, which are often treated quite differently. And there's a robust increase of undirected and decrease of directed functional connectivity, decreased information flow in the brain, and this is kind of important when we're looking at brain dynamics generally. Now, in the last bit of the talk, I want to just move away from the psychedelic compounds and talk a bit more generally about hallucinations. We can also use strobe lights to generate hallucinations in the brain. So this is a system, it's called the Lucia strobe light. It has 8 high-power LEDs that flash you can put them up to 200 hertz. And if you sit in front of these with your eyes closed, then for most people, within 2 or 3 minutes, you'll start to have some quite interesting visual phenomena, colors, shapes, sometimes very complex and rich visual experiences. And we can also, so we do this, we've been doing this is still, again, ongoing work. Take a bunch of lucky students, put them in a room, close their eyes, turn the light on, and we either leave it dark or we stimulate at 2 different frequencies, and they report continuously the intensity of the visual experience. After it finishes, we ask them, we give them the altered states questionnaire and free verbal report, and we're measuring EEG throughout the whole thing, so we can look at the brain as well. And what we find is all subjects that we analyzed reported at least simple visual hallucinations, but several reported quite complex ones. And I was struck how complex they could be. One person said, I had very vivid hallucinations of faces. I could make out their individual features, what they were wearing, and I had a hallucination of standing in a corridor in a hotel. I was standing in a parking lot, couldn't see any distinctive cars. 2 gentlemen walking towards me dressed identically, wearing a white top and brown trousers. I mean, these are quite rich hallucinations going on. And if we look at the altered states of consciousness questionnaire, this just is a spider plot summarizing the responses in to the different dimensions. and comparing the strobe light conditions with previous data under psilocybin, it's pretty similar. So people are rating their experiences in a way that bears some, at least gross similarity to the reports people give on psilocybin. And the intensity of the experience is also quite high, is much higher for the strobe than the dark control. I'll skip the power thing, but just to show you again that however you measure it, this levels of entropy goes up in this state too. So we see an increase in entropy when the brain is being perturbed by this strobe lighting that's generating these visual phenomena. This is over occipital cortex, so this is over the visual part of the brain. And I won't go into exactly what that graph means, but basically we're trying to do different ways to make sure that the increase in entropy that we observe is not totally explained by the changes in power, because you smash the brain with a strobe light and you see a lot of changes in spectral power as well. So this is just, it's a minor study, but just trying to bridge the gap. But an advantage of doing this is, of course, if you give somebody LSD or ketamine or whatever you want to give them, you're changing a lot of things in the brain. Serotonin receptors are spread all over the place, involved in multiple different things. So in a way, it's quite nice to have a model of hallucinations that does not involve drastic neuropharmacological changes throughout the brain. We can maybe, it's another comparison where we know we're not confounding things that way. That's the motivation for doing this. And that's also a motivation for the last thing I'll tell you about in the remaining 5 minutes, which is about something we built with virtual reality that is rather amusingly called the hallucination machine. And this is another way of using technology to induce psychedelic-like experiences without drugs. The idea of this goes back to the idea that was mentioned earlier about perception as a process of prediction. So imagine being a brain. You're stuck inside a skull. You've got no access to things in the world, whatever they are. All the brain has access to are noisy and ambiguous electrical signals, which could be very indirectly related to anything. I mean, they don't come labeled, this is vision, this is audition, this is to do with the table, this is to do with the mug. It's just all electrical signals. So if perception is a process of figuring out the causes of sensory signals, it has to be a process of inference. This is the Bayesian brain idea. It's the brain's best guess. What we perceive is the brain's best guess of the causes of those sensory signals. There's no... I know what I'm going to do. There's a bit of sound to demonstrate this. What this means, I think, and the sort of conceptual implications of this is, I think, still emerging, is that perception, instead of being a bottom-up process where sensory signals come in and are progressively elaborated by the brain at deeper and deeper levels, perception is as much, if not more, an active, constructive, top-down, generative process. We perceive the world from the inside out as much, if not more than, from the outside in. And here the idea is if this balance between incoming sensory data and the top-down predictions of the causes of this sensory data is altered, then we can start to have unusual perceptual experiences and we may start to have hallucinations. In this view, normal perception is just a controlled kind of hallucination, and what people typically describe as hallucinations is just an uncontrolled form of perception. But the mechanism is exactly the same in both cases. This is usually thought about in terms of predictive coding, where we have where the sensory signals now that come into the brain from the outside are the prediction errors, the difference between what the brain gets and what it expects at every level of description. And the blue arrows, the top-down arrows, carry the brain's predictions, the predictions about the activity at each level of the brain. And what we perceive then is the combined content of all these predictions of activity right down to the retina, the joint content of the brain's best guess of the causes of sensory signals. There was a very simple example of this, which I'm going to need to do here. Some of you will have heard this before, but if you have, be quiet, please. I can't do it because it's HDMI. Hold on. If I unplug it. Now.
I think Brexit
shit. Hold on, sorry. Listen to this. Anybody understand that? Okay, listen again. something. Now listen to this. This is exactly the unmodified thing.
I think Brexit is a really terrible idea.
So you can all hear that, right? And that's not a random statement. And now if I play the same thing again, the first sound. Okay, so you can probably all hear words now, and you couldn't before. So this is a very interesting phenomenon because it shows that our... Plug back in. It shows that our perception is not just the sensory data. The sensory data was completely different in those cases. All that's changed is the brain's best guess of the causes of that sensory data, and that changes what you consciously perceive. So it's about the predictions. And again, if you tweak those predictions, you might have unusual perceptions, hallucinations like this. So what we did, and I'm coming to a close now, what we did is we decided to simulate that kind of process using virtual reality and these very powerful new, not that new, class of neural networks called deep convolutional networks. So this is a deep network, many layers that somewhat recapitulate the functional anatomy of the visual system, the primate visual system. This particular network has been trained to classify dogs. Give it an image, and it will tell you the breed of dogs that are in that image. And it's very good at this. It's very successful. So the input image is processed in different ways. The last layer will tell you the breed of dog. But what you can do is you can run this network backwards, and you can sort of clamp the activity of an output unit and then change the input until the network settles into a stable state. So what you're now doing is basically changing the image to reflect the network's belief that a dog is there, even if a dog might not be there. You might have seen images like this on the internet. This is exactly the result of this process. It's called deep dream algorithm that Google developed to figure out what was going on inside these networks when they were classifying things of various sorts. So what we thought we would do is take a network like that and modify this algorithm and apply it to panoramic immersive movie. So I'm going to take you on a trip to finish to where I come from, to Brighton and the University of Sussex campus on a Tuesday afternoon. And this is panoramic video from the center of campus fed through this reverse deep dream algorithm. And basically the network is expecting there to be dogs everywhere, and there are a lot of dogs in all sorts of places. Now, given the context of this session, I don't have to remind you of what these experiences, what this video might be reminiscent of. The phenomenology is quite striking in terms of resemblance to what we've been talking about. And I think this is really important, interesting, because it's basically showing how alterations in this balance between prediction and sensory data might account for some of the very salient features of altered perception in different conditions, in this case, the psychedelic state. And of course, people wearing this would be wearing a headset and looking around themselves, and it can get really interestingly immersive after a while. You can use this technique to also simulate different kinds of hallucinations. If you fix the activity at a lower level, then instead of seeing rich hallucinations of dogs and things, you see relatively simple ones. Here we've got like features, bits of eyes and things that crop up all over the place. And lower still, and you get these weird geometric type hallucinations. So here's a way of beginning to understand how disordered activity in the visual system is giving rise to very specific features of phenomenology, both in the normal condition and in altered perceptual states. Again, we use the kind of the questionnaire, the altered states questionnaire, and we see people in the hallucination machine, again describe being in a condition that's quite similar. There's some gross resemblance to the data from psilocybin. But interestingly, when we tested time perception, now, it's a reasonably well-known result, I think, that time perception or experience of time is also quite distorted in the psychedelic state. So we wanted to test this in this as well. Are people judging durations differently? And we found a big, fat, null result. People don't experience duration differently in the hallucination machine compared to not in it. So this is also, it shows that we're not completely simulating what it's like. Of course we're not. It's a, you put a VR thing, it's not that weird. But it also suggests that the distortions of time perception in the psychedelic state might be something more to do with the drug than to do with the distortions of visual perception, because we have similar distortions of visual perception and no distortion of time. And what we're now doing, just to look ahead, is using green screen methods so we can project a person into these hallucinations. So rather than just looking passively, we can, I haven't got a video of it yet, unfortunately. We were doing this at the Science Festival in Brighton earlier this year. We can have people's virtual bodies in the scene, and we would like to move to using not just these feed-forward convolutional networks, but explicitly generative networks as well to try to map more closely from phenomenology to mechanism in these states. So to summarize this, perceptual content is the brain's best guess of the causes of sensory signals. Hallucination is a form of uncontrolled perception. And we can simulate hallucinogenic phenomenology by coupling deep networks with immersive VR without, again, like the strobe lighting, without pharmacological confound. And we see similar phenomenology, though no changes in duration perception. So to bring it all together, the first part I talked about described evidence for increased entropy and decreased information flow in the psychedelic state, which I think is very much in line with Robin's ideas about the entropic brain, but also in line with other ideas too, maybe IIT and related things. Then decoupling of directed and undirected functional connectivity, I think, is of very general significance. We didn't expect them to move robustly in such different directions. The fact that they could do and didn't have to is, I think, a very intriguing empirical data point. Finally, we've seen how we can induce simple and complex visual hallucinations with strobe lighting and see again an increase in signal diversity, and we can use the fun toys of virtual reality and AI to simulate psychedelic phenomenology non-pharmacologically. So I want to thank the various people in the lab. Again, the people named are not the people in the photo, because it's an old photo, but Michael Shartner in particular did a lot of the work on signal diversity, and of course Lionel Barnett on Granger causality, and of course very grateful to Robin and Suresh for collaborating on their MEG studies. And for those of you who were here 2 years ago, will remember Baba Brinkman doing his wrap-ups of each day's talks, which was phenomenal. The other project that got started 2 years ago with me and Robin was we discussed with Baba about doing a rap guide to consciousness. And that's now done, and if you're in the vicinity of New York in the next month, you should go see it. It's playing off-Broadway in New York after being in Brighton Festival and Edinburgh Festival. So again, thanks to Tucson for kickstarting that process, which has been a whole lot of fun over the last 2 years too. So I'll leave it there. Thank you very much.
Thank you for another fantastic talk. I'd like to welcome up our other speakers, and we will open up the question and answer session. Having moderated one of these before, I can say with confidence we all love to talk about psychedelic drugs and the psychedelic experience. Please keep your comments and questions very concise so we can have as many speakers as possible. Also, I'd like to humbly suggest that it will be more fruitful to talk about the psychedelic experience as opposed to your psychedelic experience. So stay focused and stay on this planet. Please, over here.
Hi. Yeah, awesome, amazing work. I'm super proud of you guys. One question is, do you have maybe a theory or an intuition about why psychedelic states are so emotionally charged? Like what makes valence, both negative and positive, amplified on these states?
Yeah, is this on? It is on, oh good. Yeah, there are a couple of components that I think could be candidates to explain it. One would be to think of the brain in a hierarchical way, and that perhaps the measures of signal complexity, diversity, entropy that we're seeing increase under psychedelics is something that we'll see in a very pronounced way in the cortex, whether we're knocking out organization in that most developed aspect of our brains, the thinky aspect of our brains, such that it allows a lower level of the hierarchy, perhaps the emotional system, to operate more anarchically, is one potential explanation there. But another one would tie in with Selenatozoi's recent finding, looking at our data, of increased criticality under psychedelics. And again, this property of maximal sensitivity to perturbation. So basically means if you take a system and you perturb it, if you ping it, you know, that effect is going to reverberate more if the system is in a critical state. And so, you know, emotion is something which is often comes from a perturbation. That perturbation could come from within, and often does, but it can also come from without. And so that principle of kind of highly sensitive emotionality, you know, that can be driven almost perhaps spontaneously from within, but perhaps also influenced and manipulated through things like music, for example, and that having more of an effect in a critical brain under the influence of a psychedelic.
Any other comments from our panelists? Oh, next question.
Yes, thanks for coming. This question, I think, is also for Robin. I'd like to have you speculate on what is the mechanism for the long-term effects of the psychedelics on, well, the clinical effects on depression or PTSD. Is it a decreased cerebral blood flow over a period of time? Is it affinity for the antigen for the serotonin receptor? Is it new neural networking and axonal growth, or is it something else?
I think it might be something else in the sense that, so if you go by the data, and to sort of bring it down to a more sort of human level in a way, the experience seems to be critical. And so the quality of the experience that people have seems to predict the longer term outcome. So that's a mechanism as well. You know, it's a psychological mechanism, but it likely, of course, has a neurobiological flip side. And so that question of what that is, again, is really intriguing. And I know my time management's really bad in my talks. I wish I could have gone into more depth about the criticality thing, but I think that could tie in. One thing that we found when we did the very simple but important thing of asking our patients, how do you think this worked for you? They said, well, I was disconnected from everything before the treatment, and then during it, I felt like I was connected to everything. I felt that there was this, you know, deep, profound sense of oneness. But they also said that that endured afterwards. And I just wonder whether that oneness may relate to a kind of, you know, the kind of harmony of brain activity in a critical state, that systems can nest into each other more comfortably, whereas, you know, otherwise in the world that we live in, perhaps systems are more dissonant, and that creates a kind of dissonance that we experience as psychopathology. Again, another theme I would have loved to have expanded on is this idea of you know, psychopathology in some sense being a kind of disease of modernity or an illness of modernity, that in becoming removed and disconnected from the rest of the natural world, which, you know, living things tend to exhibit criticality, perhaps modern man, you know, resides in a state of pathological subcriticality, for example. That's,
Just, if I could follow up a second, I think another potentially interesting, more mathematical analogy for this is attractor states and the dynamics of attractor states, because you can think of, you know, if you like, the pre-treatment state of depression or whatever condition it is as being in one stable attractor state. And this relates to this idea of perception as prediction. When we perceive the world, we perceive it not just as a passive inference on the way it is, but we're always selectively sampling and interpreting sensory data in many ways to align with, to fulfill our predictions about the way the world and ourselves are. So it's not just perception as prediction. Perception is often self-fulfilling prediction. Carl Friston talks about this in terms of active inference. We sample the world in a way that best supports our beliefs about the way the world is. In depression, you think about this in terms of the way events are always interpreted as supporting negative self-beliefs and negative beliefs about the world. Possibly the long-term impact of the psychedelic treatment is to knock the system into another attractor state. You don't need deep structural changes for this to happen. You can have the same structure, but it will mean it's now still doing the same thing, but because you've shifted the state transiently through this compound in such an obvious way because of the experiential change, which is why these effects correlate with the intensity of the experience at the time, then in that new state, the system, while following exactly the same process, will interpret the same data differently and actually sample the world and the self differently as well. And I think that might have a lot to do with it.
Could I add quite briefly, just that, I mean, that this idea of a sort of marriage of sort of Bayesian brain ideas and entropic brain, if you want, might chime with that quite nicely in that, yeah, as Anil was saying, you know, a disorder like depression characterized perhaps by pathological priors, a negative bias, thinking about oneself very negatively, thinking about the world and other people in this very pessimistic way, and those priors being stamped in, you know, and then if you can create a kind of hot state where you can relax or even collapse those prior assumptions, then you have a state of malleability in which you can change them. And so that would be the mechanism there.
Actually, I'd like to hear Jimo comment briefly because Jimo studied the near-death experience, which as a non-pharmacological state can be transformative in a very similar way. Jimo?
Yes, I never had a near-death experiences myself, but the animal study shows neural correlates of consciousness. We're still trying to see whether there's any relationship between near-death consciousness or conscious information processing and potentially increased level of DMT secretion in the animals, in the dying animals. That remains to be seen. I think that we need a lot more data, five more repeat experiments and to be sure. But clearly that's something that we need to look into.
But these experiences are similarly transformative in terms of value system and perspective.
And just very briefly, in our DMT work, I encouraged Chris Timmerman, who's running the studies, to issue a standard near-death experience scale. And we saw a really nice correspondence between people's ratings on this scale in the context of true near-death experiences and also the experiences that they had under DMT. So the parallels in terms of the phenomenology and likely, I think, the neurobiology are really quite strong.
Speaking of near-death experience, I'm seeing bright lights and some vague figures, but I think we're next over here.
Oh,
yeah, this is for the last speaker. In terms of you're looking for non-drug-related forms of generating hallucinations, have you thought about either migraine or optical migraine experiences?
Interesting question. Not much. So certainly there are also very interesting conditions, especially migraine, where you have a lot of visual aura phenomena, I think. The problem is it's very difficult to get that induced in a controlled situation in the lab. And we haven't studied it.
You wouldn't want to induce a migraine, but with the strobes, you might be able to introduce optical migraines.
Yeah, we try not to do that. I mean, the thing is that the strobe effect is surprisingly general. It works on pretty much everybody. We have to be very careful, obviously, to make sure we don't give people seizures this way. And so we have to exclude a lot of potential people. I think we probably also exclude for migraine for precisely that reason, but I'm not sure.
Over here.
This is a question for Anil. I'm an electrophysiologist who's also worked with these complexity and connectivity measures. I too am surprised by this decoupling that you found between the directed and undirected connectivity measures. Do you think it could be due to the fact that the power in each brain region is changing simultaneously, and that could account for the increase in the undirected?
It's a very good question, and fortunately there's a very simple answer, which is no. These measures of Granger causality are orthogonal to measures of power. There's one reason I wasn't so worried about the potential confound of power, which is not orthogonal to the Lempel-Ziv complexity, but it's not affected by power. So we're not worried about that at all. It's just that we're trying desperately to find what should be a very simple comparison data set to see this happens in another state like wake sleep or sleep dream or anesthesia or something. like that. We haven't got that far yet because this is really ongoing. But yeah, I'm pretty confident it's not just an effect of power.
And just as a follow-up, you talked about anesthesia, and as you well know, some of these measures of directed functional connectivity, symbolic transfer entropy, or directed phase lag index, appear to go in a different direction than Granger causality. And I'm curious if you have any thoughts on that. Consistently, Granger or Granger-based techniques seem to come up with disparate results, and I'm curious as to what your perspective is.
There are a lot of competing measures of directed information flow or directed functional connectivity indeed. I mean, just on one very specific, and Granger causality and transfer entropy should never go in different directions if you're doing Granger causality right, because they are formally identical for Gaussian distributions, so that should just never happen. But when you get to things that are technically a bit more separate, like symbolic transfer entropy, that will depend on how you construct your alphabet of what the symbols are. And that could potentially cause things to move in different directions. And then there are other things like directed phase lag, which there are all sorts of things. I mean, I think Granger causality is extremely principled precisely because it is identical to transfer entropy, which I think is the most sort of principled and theoretically robust measure of information flow, and the extent to which other measures deviate from that in practice, it does reveal one thing, which is the data have to be really good, and this is why we are extremely happy with Suresh's and Robin's data. I mean, beautiful data. If they're not stationary, if they're artifacts of various sorts, that can really screw things up in a way it doesn't screw up other measures of basic spectral power. These are quite sensitive measures. That, I think, explains a lot of the divergence.
Thank you.
It seems clear from all three talks that brain's structure, the phenomenal character of our experience with patterns of activity that are structured, that are sort of glued together by neuromodulator chemicals. And given that, it seems to sort of make the ordinary waking state seem a bit arbitrary in terms of how it's glued together by these neuromodulator balances. And does that put sort of pressure on the epistemic authority of our ordinary waking states? In other words, the perception and the cognition and the things that we have in our ordinary waking state are the result of this glued together patterns of activity that depend on these neuromodulator systems. And sort of a big picture question, but thanks.
Yeah, in a sense, I mean, William James said something on that this is just but one form of consciousness. In some sense, it's not that arbitrary because it's evolved this way, but the beauty of doing these kind of studies with these compounds is that they remind you in a way how important the brain is, and that it's not magic going on here, it's working on a particular neuromodulatory system, and then we're starting to get a handle on the systems levels effects as well. So that's why I think, to perturb consciousness in such a profound way as we can with psychedelics, and then, begin to get a handle on what's going on in the brain is a great example, really, of how we can sort of demystify, really, the way the brain works and the neurobiology of consciousness itself.
Hi, thank you for an excellent session, even without drugs. I wanted to know, so some of the results are really paradoxal. The entropy is increased, but the synchronization of the network, so the entropy is increased, but the synchronization of the network will decrease, right? And also the flow. So what do you think about that? How, which explanation can you give to this paradoxal, results? I think it's for the two of you, yeah.
Yeah, I mean, speaking from my thing, it's an open question. And you've got these different measures that we don't. So yes, in my case, we see entropy go up, we see synchrony and correlation go up, and we see information flow go down. How do we account for that particular constellation of results?
Yeah.
we don't know yet. I mean, there's no, I mean, we're only just trying to figure out formally how correlation and information flow should move. Even for simple systems, it's very complex. How that would relate to entropy is complex again, and then you bring in things like the predictive coding and the free energy principle, which is all about minimizing entropy, but that's only under various assumptions, and it's not whole brain entropy, it's entropy within a model of the world. There's a lot of work to be done there, for sure.
It kind of makes intuitive sense to me, really, that that this finding of a decrease in directed information flow, that's X region, the ability to predict Y region is reduced under a psychedelic. I mean, it fits this idea of the brain behaving in a more anarchic, more fragmented kind of way. It does make intuitive sense to me. And then the correlation, I mean, when we look at global functional connectivity, we do see an increase in whole brain global connectivity. So that kind of fits the increase in correlation as well. So the results kind of make sense to me.
Over here.
Thank you. A small minority of people who experiment with very potent hallucinogens report deeply alienating experiences. I've heard it described as depersonalization by psychologists. I've heard in plain English people saying, I had so much ego dissolution I couldn't get back into myself, into my personality. Jean-Paul Sartre is reported to have had some kind of an incomplete mescaline experience that led to some of the really dark parts of his existential philosophy. And I'm wondering, so basically bad trips, persistent symptoms after a bad trip. I'm wondering if you have looked at what might be happening neurologically in those experiences.
I would love to, but it's quite hard. It's actually quite hard to see bad trips during the scanning. It's quite rare to see those kind of panic experiences in general, even in the scanners. We've had a couple of examples, but when they happen, there's often some motion artifact and we stop the scan. So it's really hard to get that kind of data. We did have a protocol written to do that kind of work, but it's the cases of those so-called bad trips that we call challenging experiences, particularly in the context of the therapeutic work, because there may be something to find out, to discover when someone's going through a particularly challenging psychological experience. So I'd love to be able to address that question. And maybe it's possible through doing a study perhaps with lower doses and perhaps not enriching the environment in the way that we do in our therapeutic work, so not having music, perhaps not having the level of psychological care, but then you question the ethics of that. And you also question the ethics of doing that and the safety of doing that when you give high doses. So you'd have to give lower doses. It'd be quite a risky study, and I also think you'd have to probably use an imaging modality that's more tolerable than, say, fMRI, so something like EEG or fMRI. I think it's possible to tackle that, but it's just so hard to do, and potentially a bit risky to do as well.
Sir?
I have a question for Jimo, and maybe I should wait until she returns.
I think she's going to be right back. She had to address some urgency. Stay there. We'll see if she's back. Please, ma'am.
My question follows the previous one, and it may be redundant. How often do you have an experience where someone has a bad trip?
Yeah, it's quite rare. I'm glad people bring this up because there's so much stigma around psychedelics, and if you look at the systematic analyses of their safety profile, they're often right down the bottom in terms of a scale of harm, and yet drugs that are so normalized in our society, like alcohol, are right up the top, So this stigma is just, it's fueled by misinformation, and it's given people this really warped idea of these compounds. If you look at population study data, there's a really nice paper on this by Hendricks et al. Some 200,000 people sampled in the US. They found that psychedelic use was an anomaly among drugs of potential misuse, in that those who'd used it had better mental health outcomes, lower rates of suicidality, lower rates of psychological distress. So we really need to rethink our perception of these compounds, because they're not the horror drugs that people think that maybe they are.
Jimo has returned.
Thank
Sir.
you.
Wonderful. So, Jimo, I know your work on VMAT1 and PMAT is recent, but I'm interested in allele frequency differences in the human population and whether you've looked to see whether there are different forms of these genes and whether those forms correlate with differences in normal or abnormal conscious experiences. I remember years ago there was a report about VMAT2 and its correlation with spirituality scale differences.
Oh, it's a really good question. I have to tell you that this model was developed in such a hurry. As soon as the reviewers came with those comments, I responded and I wrote a review article. I submitted it within just 2 weeks of time and came with the model. And then subsequently we did the experiments, actually confirming the model. So I still have more papers to read. I'm sure I missed some. But I think that what's really striking to me is that the VMAT1 previously thought to be expressed only in the peripheral tissues. Now we know it's in the central, it's in the cortex. Mm-hmm. There are lots of human psychiatric patients have mutations in VMAT1, which is why I got so excited, because this means now we could finally go to NIH and say, here's a disease relevance of psychedelics. So we need to study it. We need funding to study this. But I think right now we don't really know what the VMAT1 is doing because no one even knew there was a neurotransmitter that transports. So for instance, when we know that bipolar disease is associated with the VMAT1 mutation, autism spectrum disorder is associated with the VMAT1 mutation. So there's actually, this is quite long. There are many papers published on that.
So we have time, unfortunately, for only one more question. For those of you who are standing up now, maybe after the session you can come up. Last question over here.
I was wondering if you were all aware of, if you could comment on the upcoming extended state DMT experiment with Dr. Andrew Gallimore and Daniel McQueen of Medicinal Mindfulness.
I don't think it's happening, to be honest, because we've been talking to Andrew. If it's the idea of continuous infusion and the model that he worked with Rick Strassman, so we're collaborating with Rick Strassman in our DMT work at the moment, and we are looking into how to do that modeling properly. So it's something
going forward.
Sorry?
It's going forward.
Oh, no, yeah, I'm talking to these guys, and we're collaborating, unless he's doing something underground, or he's doing something with another team. I don't know, but yeah, no, but with Rick, I think Rick would have told us, and Gallimore's often copied in our exchanges, and we've got a meeting coming up where, with a pharmacokinetics expert, we're going to look at how to do this modeling properly. I'm aware of the paper and the idea, but I don't think they've started doing the research.
We're meeting in September.
Sorry?
We're meeting in September.
No, sooner than that.
No, we are.
Oh, you're doing it?
With them, yeah.
Okay, so are we. Great.
Thank you. So we're unfortunately out of time. Please join me again for a round of applause for these wonderful speakers.